This is one line of works that I've been developing through the years, and lately has taken a turn for something interesting. And so I'll be happy to discuss that with you. So without further ado, let's go through the outline. I will present a little bit of an overview of how information, molecules, and chemical reaction can be summed up together into an information theoretical molecular communication channel. And then we'll go through studying living substrates and understanding information in them. And then finally understanding how we can engineer information through living substrates and what are the main milestones to achieve that. And then we'll go through some conclusions. So molecules and information, why can we put together these two different things? First of all, where do we take molecules from? Well, the reality where we live in is actually a mixture of many different substances. And if we were able to extract, and in some cases we have it, one only molecule type, and we put together in an ensemble of molecules of the same type, we would obtain what we call a substance. In a substance, a single molecule is actually the smallest identifiable unit that retains all the characteristics of the substance, so the substance identity, its chemical and physical property, and the ability to take part in chemical reactions. So if we were to correlate information with molecules, for sure, our atomic structure is a single molecule that represents all the reality that we live in. Or represent one of the atomic pieces of the reality where we live in. And indeed, a molecule has nanoscale dimension, and that's why we call molecular communication also a nanoscale communication paradigm, and the same for molecular information theory. So if we were to put together molecules, chemical reactions, which is a way to transfer information between substances and between molecules, and the transport of the molecules in the space, we would have all the characteristics to call this a communication channel. And through classical communication theory, we would be able to define what is a transmitter, what is a channel, what is a receiver, with source information and the information that goes towards a certain destination. And we can also call this a molecular communication link, and then from here define networks. So what are the benefits of studying such systems through a communication theory paradigm? Some examples of future application to realize networks that can perform biosensing, so transpose our sensor networks into a molecular communication environment. Simulation of actuation on biochemical processes, even inside the body, and that's why one of the applications actually I'm personally working on of molecular communication is in medicine. And finally, augmentation of the functionality of the human body as a support to implantable and wearable devices, and in particular, to create connections among them using molecular means, which in a way might be more natural and biocompatible than other kind of communication channel technologies. So information is in molecules, but information is also a very defining characteristic of life. And so cells, which are the building blocks of life on Earth, have evolved to manipulate information in biochemical environment. We know very well that DNA is not only a structure, but it has a semantic meaning because it contains information. It contains information that today we know how to parse, even if partially. And so we know how to recognize statements inside the DNA. We know how to recognize information that then is perpetrated into what we call the messenger RNA, and then it becomes a protein. A protein enables information to also have a function. So it's actually a runtime information in biology, if you want. And information is also exchanged between cells. So it's not only inside cells, but also in the way the cell understands the environment. And also it can be exchanged between cells. For example, a living tissue in our body is also an information network. Is also a communication network per se. So if you take a biological cell and you consider all the chemical information that the cell can actually attain from the environment, and you also consider communication channels that are within the cell, such as the one that creates proteins starting from DNA, or another one that is depicted here, which is a signal transduction channel that takes information from the outside environment, chemical information, and also physical information, and transduces that into a message that is meaningful to the cell to elicit a certain response. And you take into account that there are many cells immersed in this environment receiving and sending information. You get a sense of what happens in tissues. And information, for example, drives cell differentiation in tissues, drives coordination of bacterial infection, which is a negative thing, and then drives whatever happens in our body. So our homeostasis, which is our regime state where we can live and thrive, is actually controlled by a densely packed communication network that is mainly based on molecules. Molecular information, chemical reaction, and molecular transport. Those are the main phenomena that define one of the channels that we can observe in the body. If we isolate a single cell, we can understand how the single cell can get input information from the environment, signaling molecules. For example, other cells tell this cell, you are part of the skin tissue, or you are part of this organ, you're a heart cell. Metabolites, so nutrients, oxygen, and environmental stress sources. For example, a raise in temperature when we get fever. All these stimuli elicit responses in cells, and many of them actually end up activating or deactivating DNA and its transcription and translation into the runtime information structure, which is the protein. Obviously, some information can actually stop this. So, for example, apoptosis is a very interesting phenomena in cells where one cell can tell another, now you have to die to perpetuate the species of the cell because there are not enough nutrients or because we have an external threat. So if you take a cell and you parse the network that is inside the cell into simpler networks, let's say networks that have homogeneous characteristics, you will find signal transduction, gene expression, and cell metabolism as a main parsing. And if we follow information through this network, there is a great deal of understanding how a cell works. Obviously, as a biologist would tell you, these networks are extremely complex and involve a plethora of different chemical compounds and different types of molecules. But if you really can isolate on the information flow, you have a way to understand how a cell works with a different lens, which stays homogeneous as it propagates among all these, between and inside and through all these networks. So what we did through the years on this specific topic, we tried to understand how network propagate through signal transduction until reaching gene regulation. And we also studied how information propagates between gene regulation and metabolism. I will just give you an overview of one of the results that we obtained through the years. Actually, this is a 2018 paper So what we isolated, what we focused on is a signal transduction network that interest a certain cancer. Is actually, we are talking about kidney cancer. And why we chose that is because we wanted to show an application of communication theory to medical research. But at the same time, we were also sure, we knew also that cancer is one of those diseases that actually most of the time originates from an information propagation problem. And so what we studied is what happens to a certain network, is actually a signal transduction to gene regulation network inside the cell when we have cells affected by cancer. And indeed, what we found out is that if we look at this, not from the point of view of the chemistry, which is extremely complex, but from the point of view of information flow, we can define network nodes and we can define links. And we can understand by simulating this kind of network and simulating this network both for cells that don't have any disease, so healthy cells, and cells that have a disease. So in this case, kidney cancer. What happens to the information and where I have information loss? And what we found out is that there was a node, specific node here, where I have most of the information loss. I have mostly 60% of the information that I'm missing, that I'm losing. And why do I have this information loss? Is because I have a mutation in the encoding of a protein that is on a membrane, and this protein is responsible to gather signals from outside, and especially growth factors and hormone and cytokines, which regulate cell growth. And when I have this mutation, this information is lost in this link. And when this information is lost, there is a circuit inside the DNA that is practically always closed and tells the cell to indefinitely grow. And that creates the tumor. And so this is one of the first works in the literature which quantifies using these tools information loss in a network of chemicals that is underlying cancer. So moving along, what did we learn? Well, this is a new approach to estimate information flow through a single cell, but we also developed somewhat a novel metric for medical research. Indeed, we can also apply this to other type of cell, for example, to fine control the production of biological components. We can develop novel organism ontologies. Imagine classifying cells on the basis of their information capability, and then also automatic methods to inform the reduction of wet lab experiments, which are usually very expensive, and novel strategies in the future for disease diagnosis and cure. The things that stem from this work is a research that I started with a colleague, Peter Thomas from Case Western University. So for this research, we were asking this simple question. So imagine that you have two substances in an environment, and you have cells that are living in this environment, and especially imagine simple cells like microbes. So there is if those cells have to move by chemotaxis, so they have to sense the substances and somehow reach the substance as it is needed. If I have too little information, a cell will not be able to know where is information exactly and will perish. So some individuals would not know enough. If a cell starts to have too much information, and information is costly, some individuals spend too much resources to know too much, and they will perish and not live very well. There is actually a just right information quantity from the environment that enables the cell to live and thrive without consuming too much energy. There is a great deal of work in the literature to study these kind of systems. And in fact, there is a lot of research that study rewards fitness versus the information cost. And so what is this how is this trade-off built, especially through evolution? But also some researchers developed a new metric to measure this trade-off that is called Gold-Kelly information, which means what is the increase in fitness resulting from more information versus the cost. And also there are a lot of studies, and I believe some of the talks today will also talk about that, on semantic information. So where information becomes not the usual information that we studied in communication theory, where we don't care what is the meaning of the message, but each message becomes meaningful. And so the semantic becomes a value in how much information a cell gets. So it's not just improving capacity of a channel, but it's also cherry picking the right information from the environment. So along these lines, we also thought, and that was our first question, that is more relevant information, so the more semantic and relevant information, always better, even if I take out the cost. So for example, does nature always optimize a communication channel given this constraint, fitness versus cost? Or if I remove the cost, I would get something different. Will that be a communication channel that is the maximum amount of information possible or not? But then we thought, well, but if it were the case, we would see the colors all at the same time. In the same way, we would perceive information also as human at the same way. There would not be diversity in sensitive perception, which actually we observe in nature. So our research actually shifted the constraint from the information cost to the capability of an organism to actually manipulate the information channels and still try to cherry pick the information that is important to the organism. So we developed a model. This is a computational model of a multi-species in a monodimensional environment. We have two nutrients, A and B, that are essential to the organism. So the organism has always to have these two nutrients. If not, it will die. It absorbs A and B concentration from the environment and builds those two reservoirs that you see here. If any of those reservoirs goes to zero, the organism will perish. Can divide only when both stored A and B are over a threshold. So it divides only when it is able to sustain the offspring. It senses A and B gradient through chemical receptors, binomial, and moves proportionally to the steeper gradient. And finally, constantly consumes A and B. If during this consumption, A and B go to zero, A or B, there is death. So there are two possible strategies. In one strategy, we assume that the organism has receptors that are distributed left or right, and they are fixed. So it just has a fixed communication channel. There is another strategy that we consider that is adaptive. So the organism can shift its own receptors between sensing better the gradient of A or sensing better the gradient of B. And so as performance metric, we estimated the average mutual information over a population of cells at a certain time. And then the growth rate, intended as the exponential rate of growth, so the doubling rate. So the results have been actually surprising. So what you see here is the 1D distribution of A and B. We follow a von Mises distribution, which is the distribution that supports the highest entropy in a closed and periodic environment, like the one we consider. And you see here the trajectory of two different cells. This is for the channel that keeps being constant. This is for a channel that the cell can adapt. And you see already how the distribution of the cell for an adaptive follows a little bit better the two peaks of the two distribution of A and B. This doesn't tell the whole story, but this does. So if you see here, related to cell stress, which is the rate of consumption of A and B, so how fast, how hungry is the cell, you see that the adaptive strategy, despite having a higher, the equal receptor strategy, the red, despite having a higher mutual information, so the channel gathers more information about A and B location and gradient, it has a slightly lower growth, while the adaptive strategy, the blue, has a lower mutual information overall, so it needs less information, but it has a higher growth. And so we can say that the adaptive receptor allocation strategy results in a higher growth rate and correspondingly lower average mutual information than the equal reception allocation strategy. We also measure what happens indeed to the mutual information to the single individual. And we found out that the variance of the mutual information is 0, of course, because the channel is constant for the equal receptor strategy, but for the adaptive, it actually supports a variance. And so we call this variation in the perception of individuals or subjective information. So what did we learn? That maximizing information efficiency can result in a lower growth rate. Evolution can enable diversity of perception, which is really interesting. It's a new dimension to understand information in living substrate and how to model and sense it. It's a way also to think about novel bio-inspired models for the design of novel communication systems in engineering, and is a push to revise information theory to address life, because indeed information and life have emergent properties that we normally do not observe in our synthetic systems. So a little bit of a foray into engineering. This is part of a project that I've been working on since some years until now. It's still open and running. And so what we develop here is the first way to understand how a channel between two cells can be evaluated in terms of mutual information and achievable information rate, and also how we can tweak this channel using the tools that synthetic biology gives us. So how can I reprogram the DNA to actually use this channel as if it were a normal communication channel that we study and we engineer in communication theory. So a little bit of an overview of what is around in biology. So there are many systems and many ways to observe that cells actually exchange information, and it has been experimentally demonstrated many times. But what was lacking, at least at the beginning of our work, was a little bit of forward engineering tools. For example, understanding the achievable information rate, but also understanding how much can I tweak this channel and can I use it for other purposes. And so we built a system model where I have two cells, a bunch of chemical reactions inside, and the transfer of information using chemicals that can cross the membrane. This model allowed us to think about how much information is delivered in bit per message between these two cells. We used a model from biology for which we actually had all the chemical reaction rates. And so we could actually build a simulation-based analysis using MATLAB Sim Biology. Here is the system model. Here are all the equations that we used. And here are all the parameters that we took from different literature. I'm sorry, I don't have but each one of the reference corresponds to a paper. We found out that the output distribution given a uniform input distribution of the channel is actually nonlinear. And we observe a part of the linearity here, and then a saturation. So the main idea was to study the achievable information rate of this system, taking into account that it's non-linear, no smooth, and we don't have an analytical solution. There are many ways to study such a channel. For example, Blautari-Moton algorithm is one way to estimate analytical expressions out of simulation-based or anyway data about input-output of the channel. Or we propose actually another method that is iterative and is based on the Nelder-Mead optimization. We compare and validated our results with another method from the literature that is called SLEMI, and SLEMI, and is based on the Blautari-Moto. So what we did is, with our methodology, we used the Nelder-Mead optimization to actually iteratively compute the achievable information rate with no need for analytical expression of the distributions that are involved. What we found out is that our method is a little bit better than the SLEMI to estimate, especially a low SNR, the achievable information rate of this type of channel. And it's well within the two bounds, the two well-known McKellips, lower and upper bound. And so we also found out that we also found out what is the achievable information rate for a system like that, and I would I invite you to take a look at the publication for that. And then we ask ourselves, can we apply coding theory to achieve this performance? And then we have another work that we developed, which I won't talk about today, but for which we actually had a paper award, that runner-up paper award at InfoComm. So as a conclusion, we presented a computational method to estimate the information exchange performance of cell-to-cell communication. We look at the feasibility of communication theory-based forward engineering for synthetic biology. And we obtain a proof of concept design methodology for utilizing biological circuit components to design coding functions regarding cells. And we also observe through our work that biological circuits are extremely versatile in implementing functionality in the molecular domain. I just want to take a stand to thank also all my colleagues who helped me in this journey, and especially to organize in 4 years in a row an iGEM team in my university. And it's been an amazing experience that gave me a lot of ideas for my research and also formed some of my best graduate students who are working with me now. This is a thank to many of the people who work with me and many of my funding sources. And thank you for your attention.